Hi, everyone. Welcome to our Tuesday get-togethers on the cerebellum, and I am really pleased and honored to have for you Christian Hansel. Christian is a professor of neurology at the University of Chicago. He started his career at the University of Würzburg in Germany, where he studied biology, and then he did zoology at the University of Zurich in Switzerland. Then he came back to Germany at Frankfurt, where he was a student of Wolf Singer, studying LTP and LTD, basically learning and plasticity in the pyramidal cells in the rat text. He continued his interest in learning, but switched to the cerebellum when he did a postdoc with David Linden at Johns Hopkins, where he looked at Purkinje cells and he looked at plasticity of the climbing fiber synapses onto the Purkinje cells. In 2000, he got a faculty position at Erasmus University in Rotterdam in Christy Zeu's group. And then he returned to the United States, where in 2008 started his lab at University of Chicago. And this is his 15th year there. And I guess it's a celebration of that 15 years that he just had a wonderful new paper demonstrating this fantastic work of understanding the nature of organization of the Purkinje cells and their dendrites and the inputs from the climbing fibers, which he's going to be talking to you about. So without further ado, Christian.
Thanks, Reza, for the introduction and inviting me and for running this great program. I mean, that was really a super nice addition to the cerebellar community. Hello, everybody. So I don't know exactly who is on here, but I know it's beyond the Hopkins community. So it's really great to give a seminar in this format. So I would like to talk to you about Purkinje cell morphology and climbing fiber inputs in the context of these recent findings that we published, that the human Purkinje cells are somewhat different. And I'll get to that in a minute. I want to start showing you here a mouse Purkinje cell. And that is pretty much what I hope, or I think, I assume that that is the default picture of a Purkinje cell that you would have in your mind when you think about a Purkinje cell. And that is because for decades, the cellular physiological work on Purkinje cells is done in rodents and so mice and in the past in rats. And you would pretty much imagine here like a stem of the primary dendrite, and it branches higher up in the molecular layer. It would look like this cell here. So it has a very impressive dendrite, but the primary dendrite branches up at a certain distance from the soma. Okay, yeah. So this comparison across species, rodent cells to human cells, is of course not something that is only occupying us in the cerebellum. That's also something that's relevant for other brain areas. There are a few studies that looked into this in the neocortex. Here you see a data set from a study that was published not too long ago in 2021 in Nature by Mark Harnett's group at MIT. And you see here there are 5 pyramidal neurons in different species, a range of species. Here it's a mouse, and here's a human. And what you see, basically, it was a study also pointing out some electrophysiological differences. There's always something about voltage-dependent calcium channels, so on and so forth. Depending on your perspective, that can be seen as very meaningful or a little disappointing. It's always a little bit something. Does it really change your worldview? Is a question of, I guess, where you start from. But if you look at the pure cellular morphology here, I think we can all agree on that. Not really much is changing here. In particular, if you go a little further, if you look here at the differences between, let's say, a rabbit or a guinea pig layer 5 pyramidal neuron to a human one, that is not much of a big change. Here we see the apical dendrite, here the tough dendrites, basal dendrites. And I would even argue there's not much of a difference from the mouse onwards. It's just basically an extension. So the length of these dendrites, there's an expansion to double the size pretty much. Otherwise, it is remarkably similar. If we are now doing the same in Purkinje cells in the cerebellum, at first we see the size effect, of course, too. So here on the left, we have Purkinje cells from a mouse. Here on the right, from a human. Here shown at same scale, so 50 micrometer scale bars, here the same size. So it's getting bigger, it's getting way bigger. But in addition to that, there are also some additional morphological principles that change a bit. There's more change than in those pyramidal cells. It might just be a result of that we start with a higher complexity here, and this complexity is just expanding a little further. So there are no miracles happening here, but the complexity is just getting a little bit richer. So, for example, we see additional dendritic motifs that are not really there in the mouse Purkinje cells, like for example, here these age motifs here that we see in some of these human Purkinje cells. There are different things that we try to describe now better, but one thing that we focus on now, and in this last study we focused on, is what happens to this primary dendrite right in the first branch aspect of it. And then you see the difference here that very typical for the mouse is that, you, like I said before, you have this stem and then branching up higher up in the molecular layer. And here in humans, you have very often the situation that the branching happens right at the level of the soma or shortly thereafter. And then you get this, it's basically like opening of a book. So the dendrite opens right away, creates more space, and then further branching is happening right here. So now I would like us to focus a little bit on the synaptic inputs, so parallel fibers, climbing fibers, to get a little bit of starting point here, what the consequences of this might be. So in the pyramidal cells, to give us in the cerebellar field a little bit of moral boost, because otherwise we feel always a little bit inferior. I feel, but these guys here are receiving about like 15,000 maybe excitatory synapses in the human, because the density is going a little bit down, maybe 20, 25,000, maybe up to 14,000. I don't want to fight about numbers here. In the mouse, the conservative numbers about parallel fiber inputs, and many of them are silent too, would be 40,000. Less conservative, but still in a realistic range, would be 150,000 synapses. In the humans, just by extrapolation, we played a little bit around with that, and we reached numbers of conservatively 500,000. A little bit less conservatively, but not totally crazy, would be 2 million parallel fiber synapses. So we reach totally different ranges of numbers here, and all that needs to be integrated by these individual cells, which is quite interesting to think about. Now, before we go into these human cells and the aspects we studied in this latest publication, I would like to talk to you a little bit about an aspect of climbing. There are many new aspects of climbing fiber signaling that are studied, but I would like to talk about the one that we studied, of course, in my lab, and we focused on, and that is the instructive signal in parallel fiber plasticity. Because in recent years, there were several updates, and I want to use this chance to get us all on the same page and show you how delicately tuned and regulated this climbing fiber contribution to plasticity really is. And I start with an aspect that you might find surprising, and that is actually what do parallel fibers do without the climbing fiber, and then we get to what they do with the climbing fiber. Parallel fibers on their own behave like excitatory synapses in other systems too. It's not that they are just, because, you know, we all have the notion that in the cerebellum synapses do a lot of crazy different things. Often reverse, something is really different, but the synapses themselves here in the parallel fiber system actually do a lot of things that are very, very similar to what you would find in hippocampus or neocortex. You feed activity into them and they show synaptic potentiation, they show intrinsic potentiation. I just want to briefly show you that, how that works together, so we are on the same page with this. This is an unpublished study in vivo with 2-photon imaging, and the driving force behind that is postdoc Ting-Fen Lin here, and at least here in my arrangement, behind our figures here is student Silas Bush. There will be a figure of him, a picture later on. And what we do here is actually both with, it's a receptive field study with air puff stimulation on the front paw or with direct parallel fiber stimulation. We activate the parallel fiber input, we stimulate it, and we study receptive field plasticity. And we have two different mutants to separate here a pure long-term potentiation at the parallel fiber input from an intrinsic component. So there's no climbing fiber here involved, because I want to show you first what the parallel fiber does without the climbing fiber. But we want to dissect here what is an intrinsic amplification component and what is an LTP component. So the mutants that we use here to isolate or to remove the intrinsic component is a knockout that is Purkinje cell specific for the calcium-dependent small conductance SK2 potassium channel that we found in previous studies to do a lot of the intrinsic plasticity in Purkinje cells. And this is a pretty complicated CAMK2 mutant. It basically is about the intrinsic, about the inhibitory autophosphorylation of CAMK2 at threonine 305 and 306. And actually, CAMK2 is needed for long-term depression at these parallel fiber synapses. So this one actually blocks long-term potentiation because it shifts LTP and LTD in favor of nothing or long-term depression. But for you now, it's important, these ones here, the SK2 knockout blocks intrinsic plasticity, and we have previously shown it leaves synaptic plasticity totally intact, at least in slices it does. Here, this CAMK2 mutants block long-term potentiation, but it leaves intrinsic plasticity intact. And now, this is our two-photon arrangement here. We are looking at Purkinje cells in CRUISE-1 and CRUISE-2. Purkinje cells express the calcium indicator GCAMP6F. And you see here what we are doing here. We are stimulating parallel fibers. These stripes here are the Purkinje cells, these dendrites filled with GCAMP or expressing GCAMP6F. We are still stimulating the parallel fiber input, and you see the stripe here. God forbid I will mention the name beam. The parallel fiber beam is activated here. And here you see a test pulse here, which is like a 100 Hertz pulse of 8 pulses of the parallel fiber input. So nothing, and then perpendicular to these climbing fiber stripes in this sagittal plane, you see this activation of the parallel fiber input, which activates pretty much a large portion of the dendrite here, and then it subsides again. And now, to make sure we are catching the parallel fiber input, we are increasing here the parallel fiber activation intensity. We are getting this linear increases here instead of all or none. So it's really the parallel fiber response. And in the mutants here, it's the same thing. So it's just a test. It's a linear increase. We are getting the parallel fiber response. So now, this is the plasticity here. In the wild type control, nothing is happening. It's all stable. If we apply tetanus, that is, it's a 1 Hertz 5 minutes protocol, so Masao-Ito style pretty much. We see so we focus here on Purkinje cells that are responding. That's a little confusing here, but that had a different purpose. So let's focus on this middle square here. You see an increase in the response of this calcium signal measured in the dendrite. Early, late means 20 minutes, average over 20 minutes, and then late 20 minutes. So it's really over 40 minutes in total. And then we see that the signal increases over time, and it increases significantly over the wild type control. And then we look at the LTP, the long-term potentiation, and the intrinsic component. And you see here, to start with the LTP down here, it's significantly lower when there is no LTP. But there is actually, there is actually a significant component remaining. So something else is still there, even if there's no synaptic plasticity. But if we block intrinsic plasticity, is all totally gone. And the idea here is, with this activation and this receptive field plasticity, we are driving the pathway. Like all other synapses too, there is potentiation in most cases. There is an increase in the activity of the pathway, both synaptically and intrinsically. And our model here is basically, you get this synaptic activation here, but there's this intrinsic strengthening here. And we are developing this model in the neocortex and in the cerebellum that actually you need this amplification here. because if you don't have it anywhere here where the EPSP would propagate towards the soma, you would not, regardless of strength of the synapse here, your LTP can be as strong as it wants to be. If you don't have the intrinsic amplification here, there's no strengthening of this somatic output here and spike output here. So that's our dual component of synaptic, of cellular learning hypothesis we are developing in the cortex and in the cerebellum. But here I'm just showing you these parallel fiber synapses are not just because they're in the cerebellum, they are not just crazy doing something different. They're just like normal synapses, and they're behaving as such. Now we are introducing the climbing fiber to it and see what they are doing. So as you know from the field, the cerebellar field, climbing fiber co-stimulation leads to depression. So this is a pretty old study now, by now almost 20 years old. This is in slice. We would apply parallel fiber stimulation, only get our potentiation. We can saturate it here, but now if we co-stimulate the climbing fiber input, then all of a sudden we get a depression and we can reactivate the potentiation again. And we know these two signals differ. We can, I just show that here in a very simple way with camera imaging from a slice. The parallel fiber signal is very, very localized. And some of you will know that if you stimulate the parallel fiber input in a slice, for example, and if you stimulate in vivo too, just localized enough, you can barely find the calcium response. But if you co-stimulate the climbing fiber input, it's all over the place. It's in the entire dendrite. And this makes a difference for this phenomenon here. So that leads to this inverse calcium threshold curve here, where more calcium is required for long-term depression than for potentiation. That's different from the neocortical and hippocampal scenario, the BCM, like Bienstock-Cooper-Munro curve, where it's just the opposite, more calcium for potentiation than for depression. So now you're asking yourself, so why is he telling us that? That's kind of pretty old news. I'm telling you that because in recent years, there were two very significant updates that we made to the simple model that I described here. And I just want us to be on the same page with what the climbing fiber does before we really go into this human stuff. And these updates are very important about exactly this model. One is the following, and that was done, work done by former postdoc Claire Piochon in my lab. And it was about the fact that the calcium thresholds for these phenomena are not absolute. They are relative. That means it's not one calcium concentration for depression and one for potentiation, but they are shifting. These thresholds are shifting, but relatively they are intact. That means there's always, at each condition that you have, there's always a higher threshold for depression than for potentiation. What I mean by that, you see here in these experiments here, this was done also in slice. This was done with confocal imaging and electrophysiology, and we measured calcium signals in spines, and now applied these protocol typical stimulation blocks for LTD or for LTP protocols, and looked at the calcium signals. So we had a 1 Hertz protocol, meaning one parallel fiber pulse together with a climbing fiber or not, and a 100 Hertz burst. And with both protocols, repeated singular pulses or repeated burst pulses, you can evoke depression and potentiation. And you see here, blue is a single 1 Hertz protocol, if you want. You get LTP, green with the climbing fiber is LTD, orange is the burst protocol LTP, and red is then the LTD. And this is the calcium amplitude. And you see immediately there's something fishy going on here, and that is that the calcium concentration isn't quite right. They're far off. So somehow the 50 Hertz, the 100 Hertz is in the same range, and the 1 Hertz is in the same range, but they are not together in the same range. So the absolute concentration is not really in the same picture here. Here we just use a different diet to get different dynamics, different sensitivity to see. So this is a lower sensitivity. So here in the higher calcium range, we can dissect better, but the result is the very same thing. And so actually, we measured the absolute calcium concentrations here, just to give you a picture, and we came out with the 100 Hertz protocols leads to parallel fiber and climbing fiber. So the LTD condition leads to 7 micromolar in the spine. And the 1 Hertz protocol, so the 1 pulse LTD, gives you only half a micromolar. So that is the range of calcium concentrations in a spine that is still LTD promoting. And the reason for that is that the thresholds do slide. And the reason for that slide, but I'm not going into this, we did that in this study here in 2016 with mutant mice all along. I'm just showing here the basic phenomenon, but it has to do with CAMK2 and this inhibitory autophosphorylation. So basically what's happening here is you get this inhibitory autophosphorylation if the calcium signal is just high enough and prolonged enough to tease the CAMK2, but it's not long enough and high enough to stay on. And if that is the case, so it's good but not good enough, then it shuts CAMK2 off. And then you are not getting LTD because LTD is CAMK2 dependent. And for that reason, you get LTP here. So it's a threshold shift of LTD efficiently that is due to CAMK2. So that is the one update I wanted to give you, although it has been published some years ago. It's not absolute calcium, it's shifting. And that is very, very important for the interpretation of some of those results from plasticity experiments. And that's very similar, I put that here on purpose, to George Augustine's leaky integrator model, where he says, if I stimulate long enough, you get a baseline calcium level that's a little higher than normal, that also shifts the threshold for LTD induction, makes it easier to induce LTD. All that has to do with the calcium requirements of CAMK2. Number 2, that is unrelated now, but also an update that I wanted to give you, and that is what happens if we measure finally LTD and LTP under realistic calcium and magnesium concentrations. What I mean, and that's a study done more recently by former postdoc Heather Titley. Most of these studies, and those of you who are doing these experiments are very well aware of that, and slices are done with 2 millimolar calcium, 2 millimolar magnesium. That has to do that in the early days of electrophysiology and looking at calcium release from this and that, people increased these values to get more reliable effects. And then the field was just staying with that because you got reliable effects, and that was that. But then the question was like, okay, so do we see the same rules, the same potentiation and depression rules if we go for realistic calcium and magnesium? And so we tried that out and were really surprised by the results. So the realistic concentrations are 1.2 and 1. So 1.2 millimolar calcium and 1 millimolar magnesium. So the ratio is not that different. If anything, calcium is higher, but it's not the same. And what we see here, that's slice experiments. What we see here is that if you stimulate with the old concentrations and with a parallel fiber to climbing fiber delay of 150 milliseconds, we get perfect long-term depression. If we switch to this new and more realistic protocol, even if we vary the climbing fiber delay by quite a bit, we never get depression anymore. It's all potentiation or nothing. So depression, long-term depression, is totally lost. And the reason for that is if you measure with confocal microscopy, if you measure now calcium transients, we are seeing that here's a typical double pulse parallel fiber and climbing fiber protocol, and we measure calcium transients. We see that the amplitude is actually lower in this 1.2 millimolar calcium concentration, even though the ratio is higher. But just because the calcium concentration is lower, I guess the integral is lower under the curve. So this was actually a collaboration with Sam Wang at Princeton University. And so Heather Titley in my lab had before already done whole cell patch clamp experiments in rats from Purkinje cells, and we didn't get very far with that because it's very, very hard to do that. And so the numbers of recordings you get that are high quality are very low, but we got here a few Purkinje cells where complex spikes were fired. You see here an example, and we observed that complex spikes do occur occasionally in these clusters, like here 90 milliseconds apart. And then we teamed up with Sam Wong at Princeton and Mikhail Kislin, and they did some extracellular recordings, so we could increase the numbers here. And we found that in a lot of cases, actually, there are these clusters. So I'm not taking you through all that, but actually, in awake and sensory evoked conditions, we found that in a time period of 150 milliseconds, 20% on average and up to 50% in individual cases of complex spikes are in these tight clusters within 150 milliseconds. And if we now consider that for our LTD experiments here under these realistic conditions, all of a sudden we rescue long-term depression, it's back. So now here we vary, so this is now parallel fiber to the first climbing fiber stimulation delay at 150 milliseconds, but then we vary the climbing fiber to climbing fiber double intervals, and we find that at certain intervals of 50 to 150 milliseconds, long-term depression is back. So that changes now this timing curve. So if you have only one complex spike delivered, we never get depression, like I showed you before. This is near physiological situation. But if we have two climbing fibers, these are the differences in parallel fiber to climbing fiber stimulation. And then at each point, we take the best climbing fiber to climbing fiber interval. And then you see at this parallel fiber climbing fiber interval of 100-150 milliseconds, we have LTD back. So it's quite different. So when you go to physiological conditions, it's quite different. So it's an update to our own earlier data, where it's not about climbing fiber, not climbing fiber really anymore. This all or none situation that we ourselves published. It is really, the climbing fiber stimulation can be there, but it's totally ignored in a way. It needs to be a very, very specific climbing fiber signal. It needs to be prolonged and clustered. Because it seems that this ChemK2 activation, the calcium sensor activation, is very difficult. So under very specific activation conditions, only you get long-term depression. Everything else is nothing or long-term potentiation. So with that, yeah, and this leads to the scenario here. So yeah, of course, ultimately, PKC does the internalization steps. That was studied by David Linden at Johns Hopkins. But is counteracted by protein phosphatase 2A, and that is controlled by ChemK2, and that is a very important player here. So with that in mind, how the climbing fiber needs to be very fine-tuned, the calcium signal, and is ChemK2 regulated, is a little bit more complex than we previously thought. Let's return to the human Purkinje cells. So the variation in dendrite physiology here is quite remarkable, and I'll show you some examples here. So you have here these double basal dendrites, for example, but this horizontal configuration that I showed you before. There are also some with an overall vertical orientation, like this one. There are some that even have 3 primary dendrites directly coming out of the soma, like this one. Here's another one that opens like a book and leaves a cleft in the middle. So there are presumably more differences at the level of the primary dendrite or the dendrite in general that we still have to describe, but we focus here on the difference in the primary dendrite branching. Just to give you a better overview, this here shows at the same scale here, again, human in the cyanide and mouse Purkinje cells, it's the same size in green. And this also shows that actually this finding is not new. Cajal already captured human Purkinje cells, although not many examples, and also showed that there are some with the primary dendrite directly out of the soma. So we just try to quantify that now. And to quantify it, we have to categorize something here to be able to group these Purkinje cells. Otherwise, a large-scale categorization is not possible. And to do that, we went to the mouse Purkinje cells, and we grouped these in 3 groups. So one is more this Purkinje cell that we have typically in mind, so where you have this primary dendrite coming right out of the soma. Here's the opposite, poly is what we call that, where two dendrites are coming directly out of the soma, and there's an in-between stage which does a little stump of dendrite. And to distinguish that is split, these are multi-branched Purkinje cells, and this one is not, although it has two main branches, but layer higher up in the molecular layer. So to distinguish these two as categories, we say this one needs to have two soma sizes, which in mice is about 40 micrometers between the soma and the first branch, and this one is less than 40 micrometers. So this next slide shows us a little bit more schematic, where you have here's a 40 micrometers in mouse as a distinction between normative and split, and in humans, because the soma is a little bigger, 50 to 70 micrometers. So now we are diving into this last study that we had. So human Purkinje cells fall into the normative, split, and poly group. Here are the examples. And here you see mouse examples at the same scale. And now we had a tool with this categorization for quantitative analysis. So we wanted to get away just with anecdotal description of individual cells. And with this categorization, we had a tool to do that. And we studied in 3 mouse brains, 1,350 Purkinje cells in adult brains, and in 3 human brains of elderly people, 6,640 Purkinje cells. And we did that in mid-hemispheric sections. And here, this little bar graph here that you see here is basically the biggest take-home message, and that is there is quite a difference. So the normative cell is in the mouse about 50%. It has multi-branched cells, but these poly cells that are directly out of the soma is a minority, about 17%. In humans, it's quite different. It's almost universally multi-branched cells, but only 4% are normative cells, so it's almost not existent. We also see here a little bit, so that's a distribution over the lobules in these sections, and you see also here's a dominance in the humans of the multi-branch cells. There's very little here in normative cells. But what I wanted to say, there's a little bit, it's even significant, but it's not super impressive, but there is a tendency for the multi-branch cells to be stronger in the posterior lobules than in the anterior lobules. So what does that do functionally? And for this, we have to return to the mouse, of course, because the human brain is not so accessible for this study. Because the mouse brain has a minority of these cells also in the multi-branch configuration, even in the poly configuration. Here's an example again. We want to ask, like, what is it really doing, and functionally study it. And the one thing that we were particularly interested in is the climbing fiber innervation, because we had studied that in our own lab, and because it's an obvious question to ask here. And over the years, Masanobu Kano had studied this in great, fantastic detail, and he came up with this description that in development, and I copied that here from one of his papers, what happens is there are multiple stumps of dendrites here, but then one dendrite grows out. And then with this growing dendrite, out of multiple initial climbing fibers, a winner climbing fiber grows with the growing dendrite, and the rest of the climbing fibers are eliminated. It was shown in 2008 by 2 independent papers from Bosman and Conrad was one, and Otsuki and Tirano was another one, that this is a competitive situation that is calcium dependent. There's the survivor climbing fiber is the strongest climbing fiber, that's the winner, and the weaker ones are the loser climbing fibers. It's calcium dependent. It's very reminiscent of adult plasticity, long-term potentiation, and depression. And indeed, when I was a postdoc in David Linden's lab at Johns Hopkins, we showed together there is actually an adult long-term depression at the climbing fiber input. We studied that in rats P14 to 30, so it was young adult and even reaching into this developmental stage before climbing fiber elimination is complete. And here you see this long-term depression here. We found it's calcium dependent when you use BAPTA, and it's that's not shown here, but we found it's mGlu1 dependent and PKC dependent, so pretty much similar to the parallel fiber LTD. So it's really a competitive calcium-dependent situation where LTD-like and LTP-like processes in these early developmental stages decide who's the winner and who's the loser here. So with that in mind, we went on with our study here and we performed these electrophysiological recordings, like patch clamp recordings, in P20 to 66 mice in our new study. We did patch clamp recordings and filled these cells with a dye to be able to reconstruct the morphology. And we found that overall, we do that with this kind of increasing stimulus intensity and location for climbing fiber counts here. And then we found that in the total population of something like 160 cells we patched, 15% about are innervated by by 2 or more climbing fibers. And that is pretty much the number you see already in the literature, that was around in the literature. But we find here it's not a random distribution. It depends entirely on the morphology in that you almost never see it in the normative Purkinje cells, but in the split and poly Purkinje cells, just shy of 25% of those cells have double or multiple climbing fiber innervation. And to have, in addition, an independent method, a totally independent, non-quantitative method. We went for a proof of principle method, that is immunostaining. We stained for VGLO2 the climbing fiber terminals and injected a dextran tracer into the inferior olive. And the idea is here that all climbing fiber terminals would be marked by VGLO2, but the tracer in the inferior olive would only be taken up by some and not all climbing fibers. So if you have a poly Purkinje cell like this one, you might run into some examples where one side is having VGLO2 but not the tracer, and the other side has both, and that's exactly what we see here. It's a little hard to see, but you can see that in the paper. And then we wanted to see, of course, so climbing fiber double and multiple climbing fiber innervation is indeed there at these multi-branch Purkinje cells. And now we wanted to see, okay, so is there really a larger proportion? Is there really signaling independence because there are these multiple climbing fiber inputs? And for that, we went into 2-photon calcium imaging in intact mice. And that was done with GCAM6F and CRUISE-1 in adult mice of either sex. And then you see here is an example of a reconstructed poly Purkinje cell where you see these two branches coming out of a single soma. We have 3 altogether stimulus conditions or recording conditions. One is spontaneous, one is viscous stimulation, and one is other sensory stimuli. And just to show you here examples from spontaneous. So here you see recordings from 2 branches of this cell here. And at first sight, it looks pretty similar. But if you look very closely, then you see that to some degree they're doing their own thing. So a lot of the responses are across both branches, but then you also see it's quite different actually. And there are also some responses like here, where one side is just doing its own thing. And we can show that better if you subtract one branch from the other one. Then you see here it's actually quite different in this poly situation. And it's even more clear if we do the same in a normative cell. Then you see this subtraction here leads to a flat line. In a split cell, we also get a little bit more of a distinction here, although it's not quite as clear as here. And overall, we find that the local responses are a little bit smaller in amplitudes than the global responses, but they are still quite sizable. There's much more detail we show in the paper, but here's an example from the viscous stimulation. This was done in awake animals, this is done in anesthetized animals. And the question is, how much of a global response do we see? How much of a lateralized response do we see? And it's different viscous we selectively stimulate. And just to show you here a summary of what is most important, that is here, how much local responses do we see? And then this graph just shows in the multi-branched situations, we find significantly more local responses than in the normative cells. And if we measure by Z-score, unresponses or global or lateralized responses, and that includes one side lateralized or where both sides do their own thing, Then we see here, first of all, unresponsive is the biggest group in all of these dendrite classes, and that has been shown by other groups too. And then in the normative cells, we have the biggest group is then global responses, but in the multi-branch groups, we have more of these lateralized responses. Just turn off my printer, that was getting too loud. So and here, because I don't want to go through all these details of analysis that are there, we also have in the third group this multi-sensory innervation stimulation, and that means we present light stimuli or acoustic stimuli or air puff stimuli to the whisker field, and there are much more details, and that's awake again. But what I want to show you here, you find here a common theme that is quite interesting, and that is here. Here are the normative cells, here are the split cells, here are the poly cells, and on the y-axis in all three cases of the spontaneous group or the whisker group or the other sensory groups, these are measures of locality. It's a little different, but they are measures of locality. And then we put here a threshold measure, that's just mean plus 2 times the standard deviation. But the higher it is, the more locality there is. And you see that in all three groups, very similarly to one another, you see these extra peaks popping out here in the multi-branch cells, right? And that's about, so the majority of these cells, the split and poly, still do global responses, but about 20 to 30% do these local responses. So that means there is, in all these different recording configurations, there is a component of local responses that's more than in the normative group. So if you have these two branches, you get more local responses here that are putative climbing fiber responses. So finally, that's a very final thing I want to show you to stay more or less in time. The question, of course, is everything I showed you about this multiple climbing fiber innervation was now in mice that shows a multi-branch configuration that in mice is a minority situation, and in humans is universal. But I did not show you, and that's also not in the paper, I did not show you that humans are double or multi-climbing fiber innervated. After the paper was published, we obtained some more data that I want to share with you. And those data do not directly show multi-innervation, but it's getting a little bit more in that direction. What we did here is a double stain, calbindin for the Purkinje cells, and peripherin for climbing fibers. And peripherin is a climbing fiber marker that was introduced by Enrico Mugnaini in the late 90s. And you see here that it kind of very nicely stains climbing fiber inputs. First of all, it's not quantitative, like we have about 10 cells or so that we could reconstruct quite nicely. Amongst those 10 cells, we have several cases where we can see 2 climbing fiber inputs that we can separately follow and trace back into the granule cell layer. We also have individual cases of where just one climbing fiber goes into both dendrites, even in a multi-branch situation. So that exists too. Or where one climbing fiber is there and it just goes into one dendrite. But we have several cases of this double dendrite situation. The reason why I'm saying it's not definitive proof of multi-innervation is that was a point that was made by Izumi Sugihara several years ago. We don't know, we don't really know like whether what those are doing actually closer to the inferior olive, right? It could be that they are just branching very close. It's the same axon branching very close to the inferior olive or not. So since we don't know that, it's not definite proof. But we can trace those back as independent, at least into the granule cell layer here. And I'm showing you here details of this one particular one for the following reasons. So here you see one very strong climbing fiber on one branch of the same Purkinje cell, and one very weak one. You barely see this, but I pointed out with these 2 arrowheads. And the reason why I show you that is because it makes a point. And it makes a point because it's basically illustrating exactly the situation that I made here, the point that I made here. If it's really true what Masanobu Kano demonstrated here, the winner climbing fiber follows the dendrite here, the one winner dendrite, then of course, if you have now a second dendrite here popping up, directly from the soma or soon after, like here, then there might be a reduction in competition pressure and elimination pressure, and the second climbing fiber might survive. And this example here kind of illustrates exactly that, because you have a strong climbing fiber here, it goes this way, and then there was no innervation of this climbing fiber, and this independent climbing fiber even goes into this open space here and then innervates it. So it comes out of nowhere. It was this branch was up for grabs, so any climbing fiber coming in from anywhere here could have occupied it. So that makes a pretty strong case that these are really a matrix for double or multi-innervation, but it's not definitive proof. So just very quickly 2 points of what this whole thing might be good for. So before I end, so I think there could be a lot of very complex functions that this double innervation or this dendritic branching configuration could do in humans. I want to stay at a level that is less speculative and just focus on what is pretty much in front of our very eyes. And one is a pure result of geometry. So this is our classic picture that we have. So we have one dendrite here and one parallel fiber. Allow me for simplicity to use the term beam. It's not like that I believe in beams or hate them or something, but it's one package of parallel fiber input. And then there's one climbing fiber, and those two form the perceptron here and makes that happen, this associative learning machine. But now in the double innervation scenario, particularly in human cells, we see that quite often these branches are so far, like here, even in the mouse, from each other, there's a cleft in the middle. In the humans, it's sometimes, and I showed you those examples, sometimes even more obvious. And that means we are dealing with two different packages or beams or whatever parts of the parallel fiber innervation. And that means it's two different contexts of what the parallel fibers are encoding. And the answer what the climbing fibers, the double or multiple climbing fiber innervation might do, might be in that context pretty simple. They're just providing matching climbing fiber inputs, matching climbing fiber receptive fields to allow to match a corresponding, to match the corresponding climbing fiber information to those parallel fiber information contents that are delivered here, just like that. And that would mean, so it's receptive field matching, two different, that's more complicated than it should be, maybe two separate parallel fiber inputs coming in here through the climbing fibers, and you just need two different climbing fiber inputs. So here's an example from the human cells here. They can be quite separate here, and there's a cleft in the middle, right? So it's very different parallel fiber information. For that reason, you need very different climbing fiber innovation. Here is the second scenario where it's a little bit different aspect of interpretation, at least in cruise 1, 2, where you can have these different pieces of information provided. You might get a different computational aspect of interest, and that is you can bring in different modalities now. On one branch, maybe an auditory, and the other a visual input or so from the climbing fiber. You can have different qualities of instructive signals. And maybe that might be something of instructive and computational interest for some procedures that these climbing fiber input, that the entire Purkinje cell in the cerebellar cortex performs. So I leave it there, and I thank you for your interest and attention. And I hope we have time enough for questions. Thank you.
Thank you so much for that lovely talk, Dr. Hansen. We'll open up the floor for some questions now. That's okay with you. Jen raised her hand. Please go ahead, Jen.
Hi, Christian. Thanks for that lovely early morning wake up here on the West Coast.
Be careful driving.
I'm in the car again on my commute, but I'm not driving.
Okay.
Yeah. So all of the work that you described really influences my thinking a lot. But there's one thing that you said that I would like to discuss a bit, which is this idea that there can be realistic and not realistic conditions in slice. Because from my perspective, all slice is unrealistic, and yet it's quite useful. And especially the calcium. Now, I've read that, like, I don't know if you have modern papers estimating what extracellular calcium is, but I've read the ones that Bouvier cite, and they all have like a giant ion sensitive electrode and a bath above it with ACSF, which is 1.2 to 1.5 calcium. So of course, what they measure is 1.2 to 1.5 calcium. But those experiments were actually not designed to measure absolute for the most part. They were measuring changes when they stimulated. So I'm like, I don't know what extracellular calcium is, and especially in the cerebellum, Purkinje cells have like high calcium buffers. It may be different. So I just object to this idea that there's one true slice condition. And Nelson Spruceton talks about there is no such thing as the STDP curve. And what he means is that, like, there are many factors that can influence whether LTP or LTD occur, and especially in vivo, the dynamics of what's happening in the circuit for sure plays a role. And it could be that sometimes calcium is low and there's two complex spikes and you get LTD, and it could be sometimes extracellular calcium's even higher, maybe because of activity or what the glia are doing, and you get LTD with one complex spike, or the timing is like more optimal and so you get it, even if there's only one, or inhibition drops, as Jason Christie's shown. So I think we should start thinking not about like, here's the one true way you get LTD in the intact circuit, but there are a lot of factors that can influence the induction, and history, as you pointed out, like, how calcium, the history of calcium can also affect. And I think that's just a better way to think about it.
Yeah, I do agree. So first of all, it's still slice, and so the same way we have done this new LTP experiment in vivo, we should also do that with LTD. Although that was really in a totally different context. I just wanted to show that had more to do with intrinsic plasticity. Second, also, presumably all our egos are always a little bit playing, and I might have here and there emphasized too much how cool the 1.2 was. I agree with you though that when you are at 1.5 or so, that's totally okay too. 2 is presumably too high, but I just wanted to make the point that when you are
how do you know that 2 is too high? Like, if you have a paper that's compelling, I would love it if you would send it to me, because we're debating, like, okay, in our experiments going forward, how committed do we feel to these numbers that come from ACSF being bathing these measurements, the ACSF that's applied. And I don't know in vivo if what ACSF shows for calcium actually is what's there in the extracellular space.
I think the point is more than the particular calcium concentration that one can fight about. It's more that what still remains as a message is that we learn from these slice experiments in the older or the newer versions or whatever it is, that more climbing fiber activation certainly drives it more towards depression. So what the points that I wanted to make in this paper, and now showing it again, is that the window for depression is actually smaller than in some of the older works appears, because under a lot of conditions you actually do get potentiation when the circumstance for depression and the activation of the relevant calcium sensors is not optimal. And secondly, that's not the point really here in this talk, but the requirements of the calcium sensors are quite interesting too, like ChemK2, for example. Although it's an indirect molecule involved, the direct one is PKC. The indirect involvement of ChemK2 is quite interesting because it loves these prolonged calcium transients, right? And that is just something, but now the exact conditions can totally vary a little bit. That's absolutely true. So I'm not saying one thing is then right or wrong. That's totally true. Yes. And I myself should also be a little bit more, if I was ever too strict about that, I'm taking that back. Yeah. We should be, absolutely. I agree with you. Yeah. It's more tendency saying, prolonged is good for the activation, and the acceptance window for LTD is actually relatively narrow. That's actually my point here.
Thank you for that question. Next, we have a question from June Liu.
Hello, Christian. Thank you for the talk, and I really enjoyed the talk, and I particularly like the science paper you published. I got two questions. One of them is, it's kind of striking, see the last figure, the last one, you have one very thick climbing fiber, the other very thin. Well, the first question is naive, what make a axon thick? What make another axon from the same type of cells a lot of thinner? The second question is, I was wondering if you have these two split dendrites, would the activity of one dendrite on one side affect the ability of the other dendrites to be activated? Like, well, I was thinking, I guess is, let's say the thick dendrites maybe activate one branch, and then that make the second branch, the second climbing fiber, the activation maybe lower the threshold of it. Maybe there is some kind of association even between these two split dendrites. Can you comment on that?
Oh yeah, that's very interesting. So the first one, I don't know why the diameters of these axons are so extent. It does correspond, I would believe, to the strong and weaker axons that are identified physiologically, right? That would be the first thought that comes to mind. Why there are these thickness differences, I don't really know. The second part, that's a very, very interesting idea. Also because in the, and we have no data for that, but in the human cells, so I'm talking now also to people that work a lot pathologically with human tissue, and there are also, even in controls, even in human controls, there are some phenomena that are quite interesting. There are also horizontal connections of climbing fibers, and they might do a little bit of what you are describing, like having a little bit an impact from one. There's a lateralization aspect to that. So yeah, it's possible that one side can influence the other side too, and that is, we would have to see like whether any of that exist in the rodents, so we can physiologically study that. In the humans, of course, we can't. Yeah, that's a very interesting question. So far, we have no access to that, but we keep that in mind.
If you can you do it in your, when you see a split dendrite, and then if you can identify the two climbing fiber.
Oh, yeah, I see what you mean. Like just a probability thing, you mean?
Right. Yes.
Yeah. I think theoretically one should be able to do that. In some stimulation conditions, it's not so easy. For example, with a whisker stimulation, the probability of a response is very low to start with. Then you can't really do that. But there might be data sets where we could kind of try to extract that. Yeah, I can have a look at that. Yeah, that's definitely a very good idea.
Thank you.
Thank you. Do we have any more questions from the audience? Theo Rossi?
Hi, Kristen. Can you hear me?
Yes.
Yes. So hi, nice to meet you. Thanks for this wonderful talk. My question is more related to the modular organization of the cerebellar cortex. And if we consider a multi-climbing fiber innervated Purkinje cell, do the climbing fibers come from the same area of the inferior olive, or do they come from different nuclei in inferior olive?
I don't know. But I give you to make up for that.
Mm-hmm.
give you at least a little bit an answer to the projection patterns, which is not what you asked. But so the zebrine, for example, we did, and you might have had that question next. We see no correlation to the zebrine pattern. We did that in mouse and human, and there's no correlation with that. That is quite interesting. We see a little bit that's here on the right side. So that's on the left was the zebrine. We do see a little bit in the human that's the multi-branch cells that have a very horizontal orientation seem to be more in the sulcus than in the gyrus or the bank. So there are a little bit local differences. It's not a, yeah, that is actually a quite strong effect in the mouse. We don't see that really. So there's some aspects that we try to tease out here, but that is not, that is the only one that we found in addition to what I said before, that there's a little bit more of a posterior than anterior distribution. Other than that, we didn't really find anything yet. The variability is also, I think I mentioned that before, the variability is much stronger in the human and in the mouse. But then let me show you that too. We also did macaque. You see these examples from macaque, we have, but only from one brain. And they, for example, show very low variability. It's a little bit in between the mouse and the human. So they show a lot what you see where my cursor is, a lot of this here, where you see these, the split configuration, like a very short stump here and then this branch. But most of the cells do that. There's almost nothing else. So we don't see, you know, even in the mouse, we see much more of a variation. Like almost all macaque cells have that. Almost none of them has a poly cell that's directly out of the soma. And of course, we don't really know what the difference is between poly and split functionally. We have no idea at this moment. So does that mean now the big jump from functionally from macaque to human? We have no clue. But that is something we can point out here as something interesting popping up.
All right, thanks.
Sure.
Thank you, Theo. I think I briefly saw Sayed raise his hand. If you still have a question, please feel free to ask.
Yeah, I was about to ask a similar question to the last one about, so when you injected the inferior olive in the science paper,
yeah.
So this was a tracer to follow up to see basically the climbing fiber going. So is there like another experiment with two tracers, like different populations in the inferior olive with different colors?
Yeah, that could, yeah, we could theoretically do that. Yeah, we did it now just with the one because we just wanted to have a different method than just the electrophysiology. But neither, I think for me at least, that was compelling enough and informative enough to confirm that there can be two. Yeah, I mean, sure, it could be interesting to see like what the location is in the inferior olive, to see like how, because, you know, even with in these two photon recordings of the functional independence of these different branches, like the majority of the mouse cells where we think they have two climbing fibers still have a global response, right? And then of course the idea could come up, it's still two inferior olive cells, they are just like right next to one another. So yeah, that could become interesting. I still think there are so many other interesting things to do that from my perspective might be more burning, but questions. But you guys are right that that is interesting. There could be something interesting there. Yeah, absolutely. Yes. In the human though, that can't be done. Of course, for us now, it's very, very important to find ways to get more human data, right? To see like how much is there really to this double multiple innervation in humans, and that's a little bit an uphill battle at this point. Yeah.
Thank you for that.
Yeah.
Do we have any more questions at all for Dr. Hansel? All right, it looks like that was it. Well, thank you so much for giving us this beautiful talk, and thank you everyone for joining us. We'll see you next week. Have a great day.
Yes, thank you guys. That was great. Thank you. Bye.